Look, what I thought I'd talk about today was the oncogenic potential of HPV and how we can really interfere with that as a potential antiviral. So we've sort of heard today that the standard sort of targets for hepatitis B, but really why we're in the drug development business is to be able to stop the complications from persistent infection. And so what I'll share with you today is some new data from our group in Melbourne, headed up by Peter Revel, which looked at the oncogenic potential of HPV and its potential therapeutic effects. So we all know about hepatitis B and liver cancer. After tobacco and cigarette smoking, it's the second most powerful oncogenic agent out there. So how is it thought that HPV causes liver cancer? Well, obviously the REVEAL study showed higher levels of HPV replication. We've heard about the persistence of the surface antigen, persistence of E antigen positivity, truncated various weird proteins that hepatitis B virus can make, and we've got a better insight into what they are now. X variants, integrated DNA, a topic of great interest. And we know that genotype C, which the Asian genotype, is really probably the most oncogenic of all of the genotypes, but A1 and F1 can also lead to rapid HCC. But something that our group in Melbourne has been interested in for some years now has been splicing. And Peter Revel, who came to our group really as a plant virologist, his major agenda item was to actually try and unravel splicing as a mechanism of hepatitis B. And he's worked very hard to do that, and so I'll share with you some of his data today. So in HIV, splicing is essential for expression of the regulatory proteins, but really in hepatitis B, we don't quite understand what splicing actually does. Though splicing has been shown to be, or spliced RNA has been packaged, reverse transcribed, and we recognise that about 10% of the virion DNA populations, both in cell cultures and in humans, are actually spliced species. Spliced mutants are replication defective because of the way the introns are removed, and you saw from Angela's slide just in the earlier presentations that a big chunk of the polymerase that tends to get knocked out is the intron. Splice transcripts, they do not encode a pol, but importantly they can be rescued in trans by wild-type HPV polymerase. And this is why we're able to detect HPV spliced DNA in serum. Secreted spliced RNAs have recently been identified in patient serum, and Angela was actually the lead author on that in her AAC publication. And so, as I said, over the last 5 years, we've been looking at the clinical, virological and pathological significance of splice variants. So, even though it's not essential for replication, for whatever reason, HPV splicing is common during its replication and over chronic infection. And 80% of patients have been shown to have spliced HPV DNA in their serum compartment. So the splicing levels seem to change over time, suggesting a highly dynamic process. They're driven by higher viral loads. It increases at approximately 0.1% per year before development of liver cancer. And interestingly, the Asian genotypes B and C seem to splice more frequently than the European genotypes A and D. Following diagnosis of HCC, though, once HCC has sort of happened, to unravel the splicing profiles has been very difficult. Now, this is something that Angela showed a similar slide earlier, but these are the major transcripts of or the major variants that you see with splice, and you can sort of see that here is the intron here that's been spliced out, and you get these sort of splice variants that have core up this end or X down in this end. And the major one, though, that everyone's been interested in the splicing field is SP1. And most studies that I'll report today include data on SP1. SP1, this is sort of the summary of it, leads to the production of the hepatitis B splice protein, which is shown for you here. Here's the intron here, which is the donor, and the acceptor sites are marked for you. Splicing generates this 2.2 kb structure, which produces a roughly 80 amino acid peptide or protein of around 10 kilodaltons. Now, studies by Peter and also by Patrick Sisson in France have shown that 40% of patients have antibodies to this protein. It's associated with E antigen positivity and high viral load. It's associated with fibrosis, and in cell culture, it can induce apoptosis, but without cell cycle block in vitro. So the sort of questions that Peter's been asking is, how might HPV splicing be contributing to liver cancer? Recent molecular data from Dan Loeb's group has shown that splicing removes a cis-acting sequence called HM, which is shown for you here from one of Dan's papers, which promotes the relaxed circular formation of HPV DNA. And by splice removal of that particular regulatory cis-acting element, generates double-stranded linear DNA. Now, double-stranded linear DNA integrates into the host genome at a high frequency and is strongly associated with liver cancer. So this is a life cycle that I'm sure you're all familiar with. And what you can see here is that, as well as the major pathway of relaxed circular DNA, when there's failure of the plus-stranded primer through the capped oligoribonucleotide to translocate in the capsid, you get these productions of double-stranded linear DNA, which a number of investigators like Jesse Summers and Bill Mason have been shown to go on to integrate and generate a surface antigen. Now, that's just one form of double-stranded linear DNA, and splicing is a separate form of another form of double-stranded linear DNA. And this is from Thomas Tu's review. Following integration of double-stranded linear DNA, not surprisingly, the most produced open reading frame are the envelope proteins. So how might HPV splicing contribute to HCC? Well, we know that Alexander cells contain lots of spliced integrated variants. So not only are they made, but they also integrate and then are produced back again. So secreted spliced variants are associated with advanced liver disease. Patrick Soussan has shown that the splice protein is associated with increased cell proliferation and liver disease. And it's been recently shown that splicing generates oncogenic chimeric integrants in the setting of liver cancer. So with all that sort of way of background, Peter did a study with Peter Angus at the Austin Hospital, who had a liver cancer cohort, and this pilot study looked at the serum collected before patients succumbed to HCC. They extracted the DNA, and they had already optimised in a previous study with Scott Priest some years ago, before this pilot study, and developed a very robust assay for spliced DNA in serum. And they analysed 62 serum samples from patients with liver cancer, 32 of those, and 157 samples from patients without liver cancer. And this study was published recently, a couple of years ago, in the Journal of Hepatology, which showed really that, as a pilot, that indeed before the onset of HCC, certainly in the first 3 years, it was possible to be able to demonstrate spliced variants in that sort of subset of patients. And again, we confirmed the Asian data that higher levels of spliced DNA were found in Asian genotypes compared to the European genotype. But we acknowledged that this study was really a pilot study, and we needed to do a bigger study. And that led us to our Taiwanese collaborators and colleagues a little bit further north of Australia. And looking at the REVEAL study, we were able to collaborate with Professor Yan and Professor Chen. And just to remind you what the REVEAL study is, it's a 3,500 male and female participants who were seropositive for S and seronegative for hepatitis C, and they were able to identify a subset of patients who had the greatest risk factors associated with liver cancer, with high viral load, E antigen positivity, and genotype C. So in a very nice collaboration which we've just completed, we asked the question, are secreted spliced variants predictive of liver cancer in this large Asian cohort? And so here's the data. And so Professor Yan identified pre-diagnostic samples closest to liver cancer. The median time was 2.5 years from 152 incident cases, matched serum controls from just over 370 age and sex-matched non-liver cancer controls. Earlier samples from 80 liver cancers and 33 controls were also examined in this study. The DNA was extracted in Taiwan and sent blinded to our lab in Melbourne. We then did the diagnostic PCRs blinded and sent the results back to Taiwan. And Professor Yang did a logistical regression analysis, did a strong statistical study, and really the bottom line was that after adjustment for all of the other causes of liver cancer and other related risk factors, subjects with spliced DNA, if they had the level of greater than 10%, were 3.5 times more likely to develop liver cancer. But if the serum splice levels were higher in patients with older age, e-antigen seronegativity, genotype B came out in this analysis with a pre-core mutant. They were negatively correlated with serum levels of DNA and surface antigen. But really the strongest association was identified with a spliced DNA level of 20% when we increased it, and the Uden's index analysis and the odds ratio of 23 with a very strong statistical significance. So the risk of liver cancer for patients with a spliced HPV DNA level of greater than 20% raised to extremely high levels when combined with male gender, elevated liver DNA, HPV DNA, and alpha-fetoprotein, and genotype C and liver cirrhosis. So just to sort of give you that in a graphical format, almost every liver cancer patient of the incidence studies, when we went back retrospectively up to 5 years, had a spliced DNA level of greater than 20%. And that's shown for you here on the graph. So that really sort of really was quite, well, we're very excited by this result because serum spliced HPV DNA was now identified as an independent predictor of liver cancer in the 5-year lead up to diagnosis. So patients with serum splice levels of greater than 20% would be a reasonable sort of checkpoint for pre-cancer screening in this cohort. So the initial conclusions that we made with Professor Yang's group was the splice variants are associated with liver cancer and are more abundant and varied than previously appreciated. I'll come to that in a second. The mechanism is unresolved but are likely to contribute to liver cancer through increased DNA integration. So our hypothesis is like, the double-stranded linear DNA levels are increased, and there's probably expression of the novel hepatitis B splice protein because the spliced DNA assay we used was only for SP1. So what's really happening in the liver? And to sort of put this into context of what's happening in the serum. And so people are now familiar with the fact that e-antigen positive and e-antigen negative chronic hepatitis B have very different transcriptomes from the point of view of surface antigen. We know that surface antigen has a very large amount of episomally derived surface antigen transcripts. E-antigen negative hepatitis B has a very large number of integrated transcriptome transcripts. And so what that could be translating to is that, and that can be reflected in the packaging reaction. So we were of the view that it may very well be possible to sort of screen for serum splice HPV DNA in the context of a clinical preemptive diagnosis. So Angela's already presented this slide that they went on to clone and identify a number of splice variants that are secreted. And so what seems to be happening, and the point I'd like to make, is that active transcription translation in the hepatocyte is being packaged up and presented to us in the form of HPV RNA that can be identified and sequenced in the serum. So it's not too much in terms of rocket science to go to the next step and say, well, if indeed we can start screening patients at high risk for liver cancer and spliced DNA is the problem, then we can actually start to develop an intervention strategy through the RNAi interference mechanisms that Bill presented to us earlier. So we did our studies with SP1, and I think that as we go and look into the liver cancer story a bit more, we'll find other splice variants that are equally important as well as SP1. So I'd like to acknowledge Peter's group for the opportunity to present this data, and I think that as well as an antiviral program, I think people should start now looking at splicing as an important oncogenic factor. Thank you very much.